Korang ka.
Isa daw?
Hindi po po gano'n natin. Gani. Hingyan lang. Hina ako daan.
Tik-tik mo ka diya.
Pero ano lang. Char! Dili lang naman. Okay, normal lang. Okay, first time.
Kasi pagani. Sige lang. Rally.
Walk na kabal sa yung dynamic.
Kubalo si tingin dito.
Ikaw ba na?
Ay, tama ba yung istorya mo rin siya?
Kalimu lang. Sorry.
Sorry. Di masiguro. Depende. Depende kung do yun.
Hello. Morning.
Morning,
Morning,
Doc.
Doc.
Can you please close the door?
Hindi naman kasunod si Joyce.
Again, I'd like to formally welcome you to second year medicine. And congratulations to those who made it from first year. For those who didn't make it last year, again, congratulations for still being with us. Okay? So, I have to let you know the house rules during our lectures in general pathology. Number one is you should always be present when we give our lectures because we don't give a lecture every day. So you will be at a loss because you are paying a lot for your education here in our medical school. And what we're going to lecture would be basically from your book. I think it's the 10th edition. You don't have the 11th edition yet. No? So some may have the 11th edition. But again, for the meantime, we're going to focus on the 10th edition. There's just a little difference between the 10th and the 11th edition. Now, what we're going to do will be lecturing to you some of the topics in the book, but we're going to expound on it because if we lecture in detail, it will take us a lot of time. Now, we have to remind you to behave properly, you have to be in your proper decorum when inside the room, and you should come on time. Because if you're very tardy, then that would be a factor when we evaluate you at the end of the year, whether you're going to pass or not. Because this is one of the disciplines that you have to follow, especially when you become doctors 2 years from now. 2 years na lang, you will then be called doctor. Okay? Why? Because when you become a doctor, you should be respected by your, you should be respected in the community. In the past, we had some doctors, for example, in one hospital, wherein the doctor was mauled by the patient and the relatives because they didn't know he was a doctor. He was just wearing a t-shirt with yung uso ng maong with a lot of holes, parang tinamaan ng bomba, no? So when these patients are really very irritable. Why? Because they are sick and they already have a lot of these problems with the finances, etc. And if you do not act properly or talk to them diplomatically, you will get into trouble. Okay? handle patients later on. Now, when you are going to be a doctor, there is no time for yourself. You don't have the luxury of time. Remember that, okay? No matter what field you will be following, like if you become a surgeon or an obstetrician, the more so you will not have time for yourself. Why? Because in the middle of the night or while during your wedding or your birthday, your patient can come and ask for your help. So it's not a joke. Even if you become a pathologist, you can still be called to do an autopsy in the middle of the night, even if it is COVID time. Okay? So we experienced this during COVID. So we had to do the autopsy in one hospital at 2 o'clock in the morning. Yeah, you can do an autopsy at 2 or 3. And that means when you become a doctor, you devote 90% of your life to the community. Do you know that? Mahirap maging doktor. But you also have your benefits and your advantages. You easily get rich, no? Joke. No, the potential of, or the benefits of being a doctor is that the job will come to you. It's not you going to apply for that job. Okay? You will be secured. That's why we have a lot of women who no longer want to get married. Why? Because they feel they are very independent. They can support themselves. Okay? Noon, the girls, asawa ko ng kasi para safe. Now the women are very, very independent. They are now very strong-minded, especially the doctors, the women doctors. Okay? Your personality right now is very, very strong. You have women going into surgery, which was a no-no for women in the past. Surgery, neurosurgeons together. Women were usually relegated to becoming obstetricians, pediatricians, but no longer. That's no longer the story. So, when we are in this room, expect you to behave properly and to listen. If you don't want to listen, you just sleep. If you're hungry, or you just eat there. If you want to go to the toilet, you can go. But do not bother the class. Okay? So you should be now more professional because you are now postgraduate students. You are postgraduate students. You are no longer in high school. So every time that we meet, we're going to have an exam. That's a given, no? So by and by, or maybe after 30 minutes, I'll give you an exam. And that means to say that you should have read your lesson or your chapter at least 2 weeks ago. 2 weeks ago? No, because this is the way a doctor should be. You should always be prepared when you come to school or when you are already a doctor and go to the hospital. Because when you go to the hospital and you're assigned in the emergency room, there's no time to read your books in order to gauge the situation. If you're assigned in the emergency room, your mind should already be working. When you see the patient, you already know how to handle your patient. Emergency procedures especially. For example, if you have somebody who is choking to death, kasi ang kinain niya anong klaseng steak, wagyu, kobe. Yeah, we have a lot of people dying because of choking from the food. Why? Because they're talking and eating at the same time. So if you're eating and eating and talking at the same time, you may swallow the food in the wrong opening. And what procedure would you do? Heimlich maneuver. If the maneuver fails, what will you do?
Manually remove it.
Doing your throat.
Call them. Para rin daw yung strong part sa ba.
Well, parang ay.
Ayun daw, siguro i-surgically remove siya. Pero ano,
Papa sa hospital, pero scalpel.
mag-create na lang ng alternate opening for the airway para mag-insert ka ng parang tube dito sa parang yung
Sa high-end na restaurant yan, nasa saan ka ba? BGC Global. Mario's Steakhouse.
if ako yung doctor na kasama nito. Yun lang nasa isip ko din doc, yung Heimlich maneuver.
Who has a better idea? What will you do? How will you manually retreat? Yun know, laki ng kamay mo. Mamatay siya sa kamay mo. You call the waiter. Waiter! Lukong-lukong doktor. Waiter, kailangan. You ask for what? Voltaire, please. Ito ako ng reseta. Alright, you get the steak knife. Kutsilyo ng, steak. What will you do? Ikaw. Okay, ganun ka. Then you stab. You stab. Throat. Trachea. You stab it with a steak knife. That person will not die. That person will only bleed. Then, when the waiter comes, then you remove the document, then you have already an airway. That patient will bleed. So you can bring the patient to the hospital. If it's bleeding, ganunin mo lang, stop na yung bleeding. So that is one emergency procedure. Now, if you don't have what we call this a background knowledge on how to deal with emergencies, that will be a big problem. You are not yet a good doctor. Even if you have specialized in pathology, etc. But your groundwork in first year and second year, third year and fourth year is very, very important to become a good doctor. Okay? So going back to the class now. So when you are in this classroom, okay, you should always remember that anytime you may have an emergency, like a quiz. I give you a 20-question quiz by after 10 minutes. Are you prepared? Okay, very good. 25 na lang. Sige. So, what else? Do you have any questions at this point? Now, if you have any concerns regarding the department, especially of the Department of Pathology, you can go to the pathology coordinator, Dr. Marlon Marangion. If you're not satisfied, you can come to me directly. Who is the president of this class?
Wala pa to.
Wala pa. So you should elect a president, vice president, treasurer, secretary. Because all your concerns may be channeled through these people. So if you have any concerns with your grades, etc., you can come to us, and maybe we will try to help you. Okay? So, who are the medtechs here? Medical technologists. I'm very fond of calling the medical technologists. Sila nawawala. I will call their... Sila nawawala. Nag-shift course. Ano po? Kayo po. Dami dito. Okay. So, some of you are medical technologists, pharmacists. Who are pharmacists?
Manalig yung ear.
Nurses. Walang nurse.
Wala.
Engineering.
Wala po.
Education, teachers. Lab aid. Laboratory aides, secretaries. Saan kayo galing? Bio, BS Bio. Okay, so anyway, again, we're trying to teach you well, and we expect you to, give a good feedback. And hopefully, we're going to see you, not see you, congratulate you next year to reach third year. Okay? The second year last year, when I see them in the lab, they're all smiles, no? Kasi parang mayabang na, no? At this time, the women especially, do not pay too much attention to your face, no? Your hair, pabayaan mo, para kayong mga Rusyang, okay lang yan. Because when you reach fourth year, you will be blooming like a butterfly.
Sige, sige.
Maingit na yung mga ano mo.
Parang
The boys will now notice you. But this time is not the time to be pretty, etc. This is the time to read. And there should be no minute wasted. Okay? Even if you're eating, read your books. Okay? So I expect you to read your assigned chapters 2 weeks before the lecture. So that when you come and I ask you, you'll be able to answer. Okay? I know all of you are intelligent, because if you are not intelligent, you don't have any business staying here in this room. Okay? The mere fact that you are here, you have something in your brain, or maybe more. And number 2, do not waste the money of your parents. Sino nang babayad? Yung parents nyo. Hindi naman silang babayad ng tuition. They are paying also for your food, your clothing, etc. So they are very much involved. So maawa kayo sa patients. You pity your parents, no? Because when you become doctors later on and when you graduate, they'll be very, very proud of you. Now, if you pass the board exam and you get the #1 post, and say you're the top toucher, you will receive a monetary award. 500,000 pesos yata for the third, 750 for the second, 1 million for the first. Well, nice, no? 1 million. We had graduates who reached third place, fourth place, and they received around 500,000 or 350,000. So I asked them, anong ginawa mo sa pera? Namasyal ko, Dok. Where did you go? I went to Korea, Japan. So it's very exciting, no? If you reach that level. No? You don't have to be very bright or very smart. The key word there is patience. Patience. No matter how bright or smart you are, if you don't read your books, nada. Okay? So I also had the opportunity to teach at UPTGH. And one of the students was a speed reader. Yung newspaper, ginaganon. The newspaper can be read word for word in less than 5 minutes. But in pathology, na. Hours. Okay. She could not read it as fast as the newspaper. Why? Because second year is the period of encoding into your brain all the information that will be later on listed out and used for implementation. Okay? So this is the period for encoding all the information. That's why it is very difficult, because it will be very, very strange for some of you remembering all these terms. If possible, you try to memorize the whole book, even the footnotes. You will not lose anything. You will only gain everything. Why? Because all the information in the book will be retrieved later on when you reach third year, fourth year, and become a doctor. The body of a human being will never change, no? Okay, so how thick is your book now? You can memorize that. I have read that book twice, 3 times, back to back, before I took the board exam. So, I think if I could do it, you can do it. No? Why? Because every day you just read 3 pages. And then, after reading the whole book, you read it again. The next time that you read it again, it's already ingrained in your brain. So by and by, when I give you a quiz, I give the lecture, madali lang, no? Okay, any questions? No questions. So again, congratulations for being in second year, and hopefully everybody will be in third year next year. Okay, so our first topic will be the cell. And sorry, the genome. What's your idea of the genome?
The whole DNA sequences of the organism.
Can you expound more on the DNA sequences?
These DNA sequences can code for different characteristics of the organism.
Like what?
Like, for example, phenotypes such as the color of your hair or the color of your eyes.
Very advanced ka na, no? But again, let's go to the basics. Ito sabi nila, you have 3.2 billion DNA pairs, 20,000, etc., 80% of human genome. Now, basically, the genes of man is very, very complex. Okay? Why? Because even if you are twins, what's a twin here?
Severe.
Certain one, yung wife ko, may twin din eh. Identical twin, is it identical? Yung wife ko, identical din. So when she was still single, nalito ako. So both of them, sino ba? So, you know, this naman, I would just smell them, no? Because they look very identical. Yeah. Identical twins are very lucky, actually, no? Because of the high mortality rate. Is she also taking medicine? So anyway, so even if you're an identical twin, there will still be differences in your traits or your habits because of the percentage of genetic compatibility. For example, if you are identical twin, they will say that it's only 99.5, right? But if you compare yourself to the monkey, 99.9. So, parang kambal mo is chimpanzee or monkey, no? So it's very close. We're much, much closer to the monkeys than to our identical twin. And that is why when you have problems with your, let's say, paternity testing, for example, you want to know if you're really the daughter of your father or mother, you go into what? Paternity testing. That will cost you 150,000 pesos. It is a very high value examination. 50,000 for the child, 50,000 for the mother, 50,000 for the father. And it's very common among the showbiz people, no? Marami kasi mga anak sa labas, sa loob, at iba pa, no? So, but the court at this point in time would allow you a 99 point, let's say, 5% or 99.8%, and they would accept it as the truth. But in reality, remember, who has a passport here? US passport or Philippine passport, a passport to Bohol or whatever. If your name is Jose de la Cruz, and what is printed in your passport is Jose de la Cruz, without the Z, that is not allowed. they will not accept it.
It's false.
So do you see now the comparison? So you should have a 100% genetic, what they call this, compatibility. Dapat yung mother mo is 100%, 100%, 100%. Not 99.9. It's still at the moment very difficult really to have a 100% paternity testing. We have not yet seen a 100% paternity testing. So even if it's 99.9 and the father said, anak kita, there's still the benefit of the doubt. Okay? Okay, so that's the story of the genome. And we have the non-coding regions of the genome, and the critical area here would be the blueprints of the cell. You know, the blueprints is the master plan on what's going to happen or what will be the function of this gene, how will it develop, okay? Will you develop into a beautiful person or an ugly person or you become an animal or whatever? That's the blueprint, okay? Because if you don't have the blueprint, just like this building, it has a blueprint actually, on where to place the doors, the pillars, etc. Okay, so that's now a given. And we have the major classes of functioning non-protein coding sequences. It's all written here: promoter, enhancer, non-coding, mobile. Now we have here the mobile genetic elements, the transposomes. You have the telomeres and centromeres, which are usually seen in a cell that is undergoing mitosis. The telomeres, centromeres. I think you've already read a lot about this in your PS Bio or histology. You're taking some in histology? Okay. So here we have the schematic diagram of the nuclear organization of the nuclear DNA. And we have the chromosomes, you have the euchromatin and heterochromatin. How do you differentiate the two?
Euchromatin is the active chromatin.
What do you mean by an active chromatin
It's ready for transcription.
and an inactive chromatin?
The inactive chromatin has a condensed chromosome. While the euchromatin are
Talking
not
about the
functioning.
structure, you function yet. Active, inactive.
So the euchromatin is the non-actively dividing since it is functioning.
Okay, so we have here the heterochromatin, euchromatin, and it's for the euchromatin is more dispersed in its arrangement, but heterochromatin is dense, therefore it's inactive. And the end result here would be the production of a protein. Okay? Now, protein is a building block of your tissue or organ, etc. That's why you need to have protein in your body. Those people who are vegetarians, how do they look like? Payat. They don't have protein. You are not a cow, you are not a carabao, but you will only eat grass or vegetables. You should have protein, carbohydrates, and fat. Okay? Why? Because we have had a lot of experiences with people who are vegetarians and who develop what? Immune system problems. They develop lymphomas. They develop carcinomas. So you should have a diet that is a diet for human beings, not for a cow, not for a rat, not for a bird. You should have a balanced diet, okay? So for those who are trying to reduce, you can do it, but take the necessary requirement of protein, fat, and carbohydrates. So the two most common forms of DNA would be the single nucleotide polymorphisms and the copy number variations. They're all explained in your book. And you have the epigenetics, which refers to the heritable changes in gene extraction that are not caused by changes in the DNA sequences. And this is the problem that sometimes science cannot yet resolve. Okay? Wala sa inheritance. So they labeled it as epigenetics. Now, histone organization. What are histones?
Stones are the
What are they
beads
made
on
of?
the
Histones.
nucleosome, and they are wrapped with protein string.
Histones.
Histones are proteins that bind to DNA.
Stones are proteins that provide structural support to some chromosomes.
They are proteins but low molecular weight proteins. There's a high molecular weight, low molecular weight proteins. So you should be very, very specific. No? Then you have the okay. Some.
Jump chromatin. Jump chromatin.
So we are actually now entering into the realm of the function of protein material in our bodies, specifically the gene. So the histones, the chromatin, and DNA packaging. Now, what is your understanding of the chromatin material? The chromatin is that material that gives a dark color to the nucleus. Okay? The nucleus of any cell will show you a dark coloration because it is basically what, compact in its appearance. No? And DNA, what is DNA again?
DNA is, ang simple term is deoxyribonucleic acid. So, yung nucleic acid chain niya is deoxygenated. And yung isa sa mga basic pair niya is, di ba, adenine, guanine, cysteine, and uracil. Uracil po ba yung sa DNA? Thymine. To differentiate it from RNA, ang substitute ng RNA is uracil while the DNA is thymine.
Cell DNA is packaged. DNA strands are wrapped around proteins and ultimately coiled over 1,000 times. This structure is called as chromatin, and proteins are called histones. There are 5 different kinds of histones: H1, H2A, H2B, H3, and H4. Histones bind to DNA to form chromatin in the nucleus of cells. In non-dividing cells, chromatin is dispersed throughout nucleus. During prophase of cell division, chromatin condenses into visible structures called chromosomes. This electron micrograph shows chromosomes lined up in middle of the cell. The presence of histones in nucleus of cells was part of debate about which molecule, DNA or protein, is hereditary material. Of course, DNA turned out to have that distinction. However, the X-ray diffraction studies later showed histones play an important role in providing structure for DNA helix. In 1964, Morris Wilkins and Vittorio Lazzari noticed that chromatin has a repeating pattern with intervals of about 100 angstroms. This repeat is different from repeating pattern of DNA itself. Based on X-ray diffraction pattern and nuclease experiments, the 200 base pair repeat observed after digestion, which corresponds to 200 base pair of DNA wrapped around each histone core. The 100 angstrom measurement from X-ray diffraction is the width of histone core and DNA. Roger Kornberg confirmed this model through experiments. He also figured out the arrangement of histones in the core. He individually purified the histones and figured out that H2A and H2B stick together as do H3 and H4. If the H2A, H2B complex is mixed with H3, H4 complex with naked DNA, X-ray pattern for chromatin is achieved. More analysis revealed that each histone core has 8 proteins, 2 copies each of H2A, H2B complex and H3, H4 complex. This histone core with wrapped DNA is called nucleosome. This is an electron micrograph of chromatin. This fiber is called 10 nanometer fiber. But where is H1 histone? It turns out that H1 histone is not part of histone core. Instead, it binds nucleosomes to give even more structure to chromatin. H1 histone sits just outside of each nucleosome and interacts with the H1 of the other nucleosome. At higher salt concentrations, 10 nanometer is further compacted into 30 nanometer fiber. The DNA structure is already twisted. By adding more twists to nucleosomes and solenoid structures, DNA is supercoiled. Even more organization is involved in maintaining condensed chromosomes. Non-histone protein makes scaffold proteins, which maintain shape of chromosomes even if absent of histones. Up to 6 feet of DNA is packaged to fit into nucleus of cell. The DNA is wrapped around histone cores to form nucleosome and 10 nanometer fiber. The 10 nanometer fiber is further coiled to form 30 nanometer fiber, where 6 nucleosome make one turn. The 30 nanometer then looped into scaffold proteins when chromosome condenses. DNA packaging has important role in gene expression.
Okay, you have just seen the actual appearance of the DNA. These are the schematic diagram, no? But what we have just seen was the actual appearance of the picture through the electron microscope. It's a black and white thing. But again, here, the schematic diagram, you have the different colored areas to specify what would be the portion that would be represented. Okay? So you have the heterochromatin and euchromatin. They look like balls and coils each other, but these are actually the actual appearance in the body. So we also have the microRNA and the long non-coding RNA. Okay? So you have the description here of the RNA. Okay? It's a fundamental, well-conserved mechanism. Gene regulation. Regulation is one of the important words to remember. Regulate. Okay? If there is no regulation, then there will be disorder. For example, if we don't regulate your studies in second year, then you will not learn anything. Okay? So we have to be very, very specific on what to teach you and what not to teach you. Okay? Because if we overthink or we let you overthink, then you will go crazy. No? Mabubuha kayo, okay? So you have the long non-coding RNA, which modulates the expression in many ways. Both of them actually control the way that the functions of the different materials are present in your body. Okay. So we have here again the schematic diagram. And we have here at the bottom, we have the gene silencing. Gene silencing. What do you mean by gene silencing? Sometimes the genes overreact. So you have to cut it off. You have to stop it. Okay. Otherwise, some people, instead of becoming a normal person, become zombies. Or sometimes, maybe this is one of the this is one of the reasons why you have people who are autistic, people who are out of this world. And this is very common nowadays. No? You have a lot of people who need to have mental rest, especially for the Generation Z. Right? Generation Z, no? Tapos na yung mga millennials. No? The Generation Z is very interesting and amusing because you want to get things done immediately. You always want to have a shortcut. You're impatient, okay? And we understand that. No? During our time, generation, no? We were very, very patient. But now, because of the advent of science, that's very understandable for your generation. Okay? So you have here the schematic diagrams of the roles of the long non-coding RNAs, and the activation, suppression, modification, and the assembly. Okay? So activate muna, and then sometimes you suppress them, or sometimes you just modify them, and then you assemble them, and you have a sub-unit complex. So what is methylation and acetylation? What's happening there?
As for the methylation, it is the addition of methyl to
A methylation po is a functional group composed of a carbon and 3 hydrogen. The difference po with acetylation is that it's an acetyl group, so it has a carboxyl group. It has, when it comes to DNA methylation, it somehow covers the DNA so that it cannot be read by, say for example, the RNA polymerase, so it is sort of silenced.
Yes. It's like a policeman who tries to regulate the DNA. That's the basic principle there. So you can read on it, but you have to understand the basic principle or the basic function of these materials or structures. So cellular housekeeping. Imagine if this room, how many are you now? You're only 300? 100. 133, this is a big class actually. Now, when you leave this room, you will leave a lot of dust. You will leave a lot of maybe sometimes trash. And who comes after you to be the house cleaners now? In your house, you have the house help, the cleaner, the house. And look at your body. When you eat something, what happens? The food is digested, and after it's being digested, it gets out. Okay? When it gets out, where does it go? It goes into the waste disposal or the throw. So that is good housekeeping. Imagine if you just defecate anywhere, no? Dito ka sa isang kanto, while I'm having the class. No, there is some regulation. No? And you have the cellular housekeeping, and you have the intracellular organelles that has already been discussed to you in histology. Okay? What are the basic intracellular organelles? For housekeeping.
Lysosome, Doc.
What does it do?
It contains specific oxidative enzymes that can degrade maybe free radicals or
What are free radicals?
active mga oxygen species, Doc.
What does it do?
Kinukuha niya yung electrons ng mga stable na molecules so that they will become unstable.
Then what happens when they become unstable?
They cannot do their function, Doc.
Like what?
Say for example, chemicals or enzymes inside the cells na responsible for example glutathione, Doc, ganun. So, ma-deactivate niya.
Is it related to a sort of a malignant process?
Pwede po, Doc.
How?
For example, if yung enzyme is responsible for regularly checking yung rate of mitosis, so if maano siya ng free radical, hindi niya ma-regulate yung cellular activity.
Okay, so very good. So it has the function of regulating the activity. If there is no regulation, for example, in our community, wala kang traffic lights, wala kang policemen, etc. It will be a chaotic community. Okay? So you try to drive around and there's no traffic light, no traffic aid, marami mo tatapanggaan. Okay? So, you have the endosomal vesicles, which shuttle the internalized material to the specific intracellular sites or to new synthesized materials to the cell surface or to the target organelle. Okay, that's very self-explanatory. And we have the functions of the cell, 1, 2, 3, 4, 5, 6, 7, okay. Protection, nutrients, communication, movement, renewal of cells, molecules, molecular catabolism, energy generation. So they are all very, very important to the cell. For example, the nutrient, the protection from the environment, communication, movement. Imagine if there's no communication right now, how can I teach you? Okay? If there's no movement, you'll be what? Confined to your house or your room. If there is no renewal of the senescent molecules, those senescent molecules, just compare it to your skin. You try to rub your skin, anong makikita mo? Libag. No? That is not dirt. That is actually what? Dead squamous cells mixed with a little bit of dirt and some sweat and etc. And then you have the molecular catabolism to what? Process the food that you have eaten. Kumain ka ng napakamahal na steak. Ano klaseng steak yun? Ostrich steak or crocodile steak. It's very expensive, no? So you will defecate after 1 month kasi sayang yung binayad mo. Ay, nako. So, anyway, we have the energy generation. If there is no energy, then there will be no motion or movement. If you don't have energy, you will not have light. You will not have water. Okay? Everything almost depends on energy for motion, communication, etc. Okay? So you have here the different organelles in the liver cell or the hepatocyte, and their different roles in the cell. For example, cytosol, what does it do? Mitochondria, endosomes, etc. Okay. Maybe I'll ask this by line. I'll memorize it. So and then I'll ask you, what's the role in the cell? Dali lang. Can you remember that? Mamaya na lang tanong. So you have here the cell. Again, you have the basic constituents, which are self-explanatory. You have the smooth and the rough endoplasmic reticulum. What's the difference between the smooth and the rough endoplasmic reticulum?
Often the plastic reticulum is embedded in liposomes.
The rough endoplasmic reticulum contains proteins. However, the smooth does not contains proteins.
Or the rough ER, its main function is for the synthesis of proteins, while for the smooth ER, it has something to do with lipid synthesis.
Like what?
Such as steroids,
What are the steroids? Example?
testosterone, progesterones, and
Estradiol.
What does testosterone do?
it is responsible
Secondary
for the male characteristics.
Like what?
secondary male characteristics.
Deepening of voice.
How about progesterone?
Progesterone.
It's the main hormone responsible or dominant in pregnancy. Pregnancy.
Progesterone?
Sometimes responsible for
So progesterone is a female sex hormone. So basically, females, it's more on regulating the mood function. By default.
Can a male person have progesterone?
yes.
Yes.
Yes, ma'am.
Estrogen.
Yes, but for small.
Why will it be produced?
Naman di ba? Pero small amounts lang.
Mga zona reticular.
From ano siya daw? Androgen diet.
In particular for males, the estrogen progesterone can be produced in the adrenal gland, particularly in the zona fasciculata. No, reticularis.
Adrenals. From the adrenals. Now they can come from the adrenals. That is why sometimes you really want to become, if you're a boy, you want to become a girl, you go back, you go to the adrenals and you try to suppress, or you try to increase the number of progesterone, or the amount of progesterone. So that's why transgender people can look like the real thing when they do hormonal therapy. But the only thing is, even if you are very successful in being a transgender, one of the drawbacks would be your skeletal appearance. Ang lalaki ng kamay pati paa mo, which is very unladylike, sabi nila. Okay? You can never change the skeletal structure. Even the head of males are always larger than the females. Okay? But if you're so blessed that you have a small head and small skeletal system, well and good. And that's very common in Thailand. No? When you go to Thailand, you will be what you call this, mabubudol kayo, no? Because they're very, very pretty. But when they talk to you, what's your name, sir? Okay, so going back. Another structure will be the plasma membrane, which is used for protection and nutrient acquisition. Okay, so you have the presence of phosphatidylinositol, phosphatidylserine, the glycolipids, and the sphingomyelins. Okay, the plasma membrane offers, or the main function will be protection and nutrient acquisition with these different elements. Okay. So phosphatidylinositol can be phosphorylated and serve as the electrostatic scaffolding of the intracellular proteins. What do you mean by scaffolding? Like, for example, the ceiling. you have the scaffolds. You know that the scaffolds easily will collapse. And one of the scaffoldings in the body would be, in anatomy, would be the ribs, the long bones, the flat bones. They are part and parcel of the scaffolding of the human body. And so is with the cell. You have the scaffoldings, okay? Basically made up of intracellular proteins. So we have the phosphatidylserine. So we have here, it serves as a cofactor in clotting of blood, etc. Again, the protein substance is always present. Okay? And then you have the glycolipids, sphingomyelin. It's very important for cell-to-cell, cell matrix interaction, inflammatory, and even recruitment of sperm to egg interactions. So there's always that flavor. No? It's not all proteins. You should also have some lipids. Lipids. You know what's lipid?
Lipid, Doc, is one of the four macromolecules which are insoluble in water. Po. It's a large group of molecules po, which are insoluble in water. Examples include fats and waxes.
That is why if you don't want to get drunk, and you're in a party, for drinking tequila straight from the bottle, you drink one bottle of mineral oil or cooking oil, you will never get drunk. Why? Because the stomach will not absorb it immediately. Okay, the alcohol will talk in alcohol. So that's one of the tricks to remember. You don't want to get drunk, drink cooking oil. Okay, so that is lipid. Okay. Okay, now, so you have the schematic diagram here of the plasma membrane. Okay. And you have the presence of proteins and glycoproteins that are involved in ion, metabolite transport, fluid phase, etc., cell ligand, cell matrix interactions. Okay, so they are involved mostly in transport, in communication, and in the reception of macromolecules. What are macromolecules and examples of macromolecules? Example of a macromolecule.
Protein.
So macromolecule, what are the characteristics of a macromolecule?
Usually found in the body in larger amounts.
Large.
Large.
I want a specific example.
So I think for examples of macromolecules would include carbohydrates, proteins, lipids, and nucleic acids. So they are grouped according to the building blocks. So for instance, for proteins we have CHON. So for carbohydrates we have CHO. So they would be differentiated by those building blocks and shared properties.
Yeah. Very good. So you have most of the proteins are integral or transmembrane proteins. They may be synthesized into cytosol, membrane through the GPIs. And the peripheral membrane proteins may not covalently associate with a true transmembrane protein. What is a true transmembrane protein? Any volunteer? True transmembrane protein.
A transmembrane protein is a protein that is transpanning the whole cell membrane. peripheral protein that is present only in the surface. Yes.
Very good. Okay, so anyway, they look very insignificant, but later on you will realize the importance of these structures. So many plasma membrane proteins function together as large complexes. Okay? They may be either in groups or aggregates, but are controlled by chaperone molecules in the rough endoplasmic reticulum or by lateral diffusion in the plasma membrane followed by complex formation, site 2. Okay? Now,
many G protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed. And afterwards, they can either be degraded in lysosomes or activate new signaling pathways.
okay, so it is comparable to a factory, food factory, wherein the meat is brought into the factory, is processed, and the end product would be a box of canned goods, or maybe a box of bananas, etc. So you have a processing mechanism. But again, this is not perfect. So that there will be some what we call backup mechanisms to correct or to renew the regular functioning of that machinery. Now we have passive membrane diffusion, and then we have the carriers and channels, and channel proteins, carrier proteins. What is a channel protein? Channel protein, any idea what is a channel protein? And
channel proteins are transmembrane proteins which, unlike carriers, they are already open, so they allow any ions or molecules to pass through the membrane.
Okay, so they are what very good pathways for transport, etc. Now, we have the again the schematic diagram. We have a lot of diagrams here, but actually these are very useful because by just looking at the end result, you have either phagocytosis or transcytosis, and you have the roles of the phagosomes, lysosomes, and even endocytosis and exocytosis. Okay? So these are very basic, especially when we talk about the mechanisms or the principles behind the activities in your body when you get sick or when you take in medicine, what is happening, or when you take in bacteria or viral organisms into your body, what is happening, and how come that when you take in the medicines or drugs, or when your autoimmune or when your immune system comes into play, after 2 or 3 days you become well. So we have here the roles of endocytosis, phagocytosis, especially phagocytosis, because it is sort of a cleaner or a policeman in your body. It digests or kills the bacteria or the viral organisms that may have invaded your body. Okay? So the lysosomes, the enzymes, all of them are very useful. Imagine your mouth, okay? Every day, do you brush your teeth? Okay, do you wash your mouth every day? When you go to sleep, do you brush your teeth? If not, you have just eaten a lot of pizza, eaten a lot of steak. Then try to sleep at night. When you wake up in the morning, you try to kiss your boyfriend or girlfriend. Mahihilo yung girlfriend and boyfriend mo. Why? Because of the putrefactive activity there. Because of the enzymes in your mouth. Okay? You try to smell the poo of your neighbor after he has eaten a lot of meat. The smell of the poo in a high protein diet is terrible. No? It's like a nuclear bomb. No? But if you eat kamote, it's very sweet, no? Sweet smelling. No? But this is due to the nitrogen that is being produced. Okay, so anyway, the role of phagocytosis and endocytosis is very, very important in the cell. So we have also the cytoskeleton and the cell-to-cell interactions. A while ago, I already made some introductions to what is present in your cell. And this is the scaffolding, etc., which would play a big role in the cytoskeleton of the cell, as well as the cell-to-cell interactions. By these, characteristics, they can adopt a particular shape, polarity, and maybe organize that relationship to the other organelles that are present in the body. And again, they depend a lot on the scaffolding of the proteins, like the cytoskeleton. Imagine if you don't have the pillars or the scaffoldings in this building, then you are very much in danger of being killed when they collapse, especially when there is an earthquake. Okay? We have seen this a lot during the last years, wherein you see buildings topple down or the ceilings topple down because of the poor scaffolding of the roofs or the ceilings. And this is usually seen in government offices wherein the job is not properly done. No? Instead of placing maybe 10 bags of cement, they just place 5 bags of cement. Nasaan yung ibang 5 bags of cement? Nandun sa bulsa. Okay? Or the condominiums, some condominiums that collapsed. Why did they collapse? Because they used cement that was supposed to be used for the road and not for the pillars, etc. They used the cement for the road or the pathway have a lower grade quality than the cement for, we call this the pillars. Sino bang architecture student dito, or engineering? I'm not so keen with the PSI, etc. So this is what we're gonna happen if your cytoskeleton is very weak. And why is the cytoskeleton weak? Again, you go back to genetic traits. The genes that you inherited may be very, very faulty. This is very, what you call this, specific, especially for people who marry their sister, their brother, first cousin, second cousin, third cousin, etc. And this is very common among the people with royalty, okay? They develop a lot of blood dyscrasias, okay? They develop a lot of blood dyscrasias because of the genetic, we call these problems. You are not supposed to marry your brother or sister. And this is still very common in the Philippines, especially in the far-flung areas, the rural areas. Why? Because they don't, or they have a limited capacity to interact with other people. So who do they marry? They marry their sister, brothers. You look at those badjaws on the streets. They are closely related. And that's why you see a lot of abnormalities in some of them. Some of them have 6 fingers, some of them are small, some of them are tall, and some may be, they may look perfect. Okay? Naswertehan, no? Yung genes, no? But most of the time, these people have genetic problems. They are sometimes low in intellect, no? They just beg and beg. I think in the Muslim world, the badjaws are the lowest, no? Kasi yung level of intelligence nila is very, very low. And they will dive into the sea for a 5 centavo, 1 peso, and they burst their eardrums. No? So unfortunately for our Bajau brothers, okay, so going back to the cytoskeleton, the cellular actions, okay, this is self-explanatory. And we have 3 major classes of cytoskeletal proteins: actin, the intermediate filaments, and the microtubules. You have here the actin, okay, and then for the intermediate filaments, you have the lamin A, B, C, desmin, neuron. These are very vague at this point in time for everybody. But later on, when you become specialists in your field or doctors in your field, this will again come back in the form of what? Results due to what we call these results are for validation of results through immunohistochemical staining. For example, for desmin vimentin, if you do this special stain for this particular test, then we will be able to determine whether the tumor is a sarcoma, a fibrosarcoma, or a leiomyosarcoma, etc. Okay. I read this in second year. Desmin vimentin. The neurofilaments can be distinguished again through the use of immunohistochemical staining. The cytokeratins. We don't know if it is a stromal tumor or an epithelial tumor, so the doctor would just request, request for a pancytokeratin stain, and if it stains for pancytokeratin, then it is an epithelial tumor. If I want to know if this tumor is coming from the ovary or from the intestine, I would just request for a CK7 or CK20. CK7 is positive for ovarian tumors. CK20 is positive for gastrointestinal tumors. Ganun kadali. No? Okay. You will meet these topics again later. So here we have the cytoskeletal elements and the cell-to-cell interactions. So it looks like a web, parang magulo, no? But these structures that go haywire are actually very important for the cytoskeleton or for the stability of that cell. Okay. So you have the cell-to-cell interactions, and you have the occluding junctions, anchoring junctions, and the gap junctions. Can you explain to us what are all these junctions?
So for occluding junctions, we have from the name itself tight junctions. So it permits molecule movement towards.
Example. Give me an example.
So take for example, there is let's say a protein molecule from this.
In your body, what part of your body can show you or demonstrate an occluding junction?
Oh, my skin.
Skin. Okay. Why the skin?
It permits external molecules or external external molecules such as dirt to come in contact with my body, which could cause damage.
Okay. Anchoring.
So for your anchoring junctions or for your desmosomes, they attach cells. So one example in my body would likely be the tendon that attaches my muscles towards my bone. And then for the communicating junctions, as mentioned there, it mediates the passage of chemical or electrical signals from one cell to another. So an example would likely be my neurons that would generate signals from one another for communication.
Okay, very good. Okay, so next you have the biosynthetic machinery and the plasmic reticulum. I think we talked about this a while ago. And so you have again the presence of the proteins and enzymes and intracellular degradation. What's the Golgi apparatus actually doing? Yung mga sa histology. Can you please close the door? Golgi apparatus, histology. Okay, please refresh our minds.
The Golgi apparatus, they modify from the endoplasmic reticulum, then forward siya sa Golgi apparatus to process the newly formed proteins and synthesize the export the synthesized products.
The Golgi apparatus is more like a post office wherein it processes, packages, and transports proteins and lipids for secretion or when it needs by the cell.
It's sort of a processor, no? Dito ka, you go to Davao, you go to Tigos, you go to Bohol, you go to Siargao, you go to Boracay. Ganyan, no? So it's sometimes very easy to make an analogy, no? Comparison with the actual happenings in the cell, so it can be easily remembered. Okay? So waste disposal. Okay, here, I think we already talked about this. And the most important structures here would be the lysosomes and the proteasomes. Okay? For the lysosomes, we have the roles of phagocytosis, autophagy, fluid-based pinocytosis. What is autophagy?
Autophagy, Doc, is when the cell, yung cells na yung organelles na hindi na ma-use ng cells, kaya iyan ang gina-dispose, gina-degrade na lang niya, Doc, para ma-use niya ang micro parts into a useful one instead. So yung ma-degrade na proteins, ma-reuse niya again.
Oh, si Madam. May sasabi pa.
So basically, autophagy is once a cell is senescent now or nearing its done its function na it gets, in order for it to be it's close to its expiration. In order for it to be reused again, if may functional pa siya ng components, the cell basically eats itself in a form of recycling.
How does it eat itself? itself?
Sa pagkaintindi ko lang, Doc, is maglaki ang membrane and then it forms an autophagosome. It's basically a combination of a lysosome and the membrane itself.
Means to say that it will call for help. Help! So what do? You call for the enzymes. Remember, if you eat food and there you don't have enzymes to digest the food, you will die. Because when you eat meat, when it comes out, it will still be meat. Karne pa rin. So, it will block your gastrointestinal tract. And that's why you have the stomach, the small intestine, which will, process it and even absorb the nutrients. Then pass it on to the next level, the large intestine, where it will be stored for a few hours, a few days, a few months, etc. Few months, years even. Yeah, because sometimes this occurs in people who have problems with their gastrointestinal tract, especially for people who don't have adequate supply of ganglion cells that are responsible for the peristaltic motion or movement of your gastrointestinal tract. And that is why some people sometimes store the waste products in their abdomen or their bodies for 1 year, 2 years, 20 years. And their abdomen becomes larger and larger and larger. So they become walking septic tanks. And I actually did an autopsy on a patient who died because of compression of the lungs from the expanding abdominal girth. And when I opened up the patient, it was like parang volcano, lumabas yung tae, up to the ceiling, because it was there for the last 20 years. Because he had an abnormality. He had a congenital, or what you call the Hirschsprung's disease. Walang ganglion cells. It is very common, especially nowadays. You have a lot of babies. Production line was not so good, so kulang ng ganglion cells. So we have to cut the intestines without ganglion cells, or sometimes we have to support it by doing a colostomy. For the meantime, that we will wait for the growth or the appearance of the ganglion cells in the gastrointestinal tract to make it functional. Okay. So later on, some of you may not be so fortunate. You may have children or babies who cannot defecate. Iba naman yung ayaw mag-defecate kasi mahal yung kinain niya a few hours ago. Ay, sayang 500 pesos na steak. I'll defecate maybe after 3 or 4 weeks. But if you have a what you call this defect in your gastrointestinal tract, then that would be a problem. So anyway, we have here the role of phagocytosis, autophagy, pinocytosis, to help the body dispose of all the non-functioning or non-usable materials in your system. So,
lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
Waste product na siya. Anyway, sorry for the technical problem. Okay, anyway, I hope it was already clearly explained. Now here we have the diagram again of lysosomal degradation on autophagy, heterophagy, okay. Phagocytosis and endocytosis. They are on your book. And then we have the exocytosis. It's throwing out the waste products, okay. Exocytosis. So again, you have the proteasomal degradation, and the end result will be apoptosis. What is apoptosis again?
Program cell death.
Program cell death.
What do you mean by program cell death?
So basically, apoptosis means that when the cell is again very old na, it may undergo autophagy or apoptosis to protect other cells from being affected by its toxic contents.
Means there is an expiry date. Like the food that you see in the supermarket, it will expire on this day. So there's already a program when to eat it and when not to eat it. But sometimes some businessmen would replace the label, good until next year, no? When in fact it's only good for next month. So what do you develop? You develop
Apoptosis. Oh. Cell death.
diarrhea, magkalibang na. Okay. So always look at the label of the food that you're trying to eat. Anyway, so just to make you wake up, okay? So we have the cellular metabolism and mitochondrial function. There is energy generation, there is intermediate metabolism, and you have the cell death, the form of necrosis and apoptosis. What is the difference between necrosis and apoptosis?
Apoptosis, Doc, is the program cell death. Meanwhile, the necrosis po, Doc, is na abnormal siya within the body. That's why maunti-unti siya na
Ano lang ito?
I think ang necrosis, Doc, is there is an external factor, Doc, na nag-cause ng cellular injury, whereas like burns.
What are the different types of necrosis?
Different types of necrosis. Necroptosis ka to?
Coagulative and liquefactive necrosis. Gaseous necrosis, fat necrosis, gangrenous necrosis, Doc.
Coagulative. Ano ba yung coagulative?
So what is gaseous necrosis?
Tatay.
It's like it's like a ano po, ang cheesy appearance po yung ano. And it is most commonly seen in tuberculosis.
She's like white appearance. Chapter 2.
Local lang daw kasi super local.
It's like the cheese that is locally made, milk and Carabao, crumbling, marupok. Unlike the cheese abroad, mga galing sa Amsterdam, you know, tortilla, but local cheese, malambot, no? Crumbly. And that is the best example of a gaseous necrosis. Now, if you have a heart attack, like a myocardial infarction, what kind of necrosis will you have? Anyone guess?
Coagulative?
Ischemia.
Yeah. Hindi naman necrosis in skin. You have a heart attack, yeah, there is, yeah, that's true, ischemia, loss of blood flow. And what will happen is that there will be destruction of the cell, but with the retention of the cellular outline. in liquefactive necrosis, there is no longer any evidence of a cellular outline, etc. Now, caseous necrosis is a combination of liquefactive and coagulative necrosis. Okay. And that's very common in tuberculosis, but we will go to that later on. Okay, so here we have cell death and through necrosis and apoptosis. Okay, this is best explained here. The ischemia or the toxins, and then you have the ATP generation, apoptosis signals, and the phagocytosis. And the end result would be cell death or cell death through necrosis or cell death through apoptosis. Now, the lifespan of red blood cells is how long?
120 days.
120 years? 120 days. Why? What happens after 120 days?
You just have reach.
Yung pag
Spleen.
yung RBC is makatravel siya sa screen, doon po siya parang mamamatay pag kat-integrate.
Where will that go?
Yung contents ng RBC, especially yung mga iron, is parang i-recycle ulit.
How about the others? Where will they grow?
Yung naman structures is recycled for
I'm asking, where will the other parts of the red blood cells go? Nasaan yung mga medtech ko? Raise your hand, I will give you 100. Wala medtech. Where will the other waste products of the red blood cells go?
Ma-recycle lang po, di ba? Ma-recycle lang po yung mga other parts.
Baligyan niya sa second hand. It will go to the graveyard.
Spleen.
Yes, spleen. Very good. Yes, spleen. Remember that because later on when we go further in our studies, there will be a lot of diseases wherein there will be a lot of red blood cells involved. And the spleen will become bigger and bigger and bigger. What's the normal size of the spleen? Nakamagaling sa anatomy. Ito, magaling sa anatomy. Normal size of the spleen. Who knows? Hala. Madam.
Ganito.
14 cm.
14 by... Give me the full dimension.
How long siya daw is 11 to 14 cm.
11. Ang width. Sige na lang.
6 to 8 cm.
The length around 10 to 11 by 5 to 6 cm.
11 by
4 cm. And it weighs approximately around 300 grams. And the cut surface will be a reddish soft surface because of the formation of blood. And histologically, you have the white part and the red part. Right? Good. Now, let's continue. Cellular activation. Okay? You have cell communication is very, very important because if the cells cannot communicate with each other, then your body will be topsy-turvy. Remember that when you see food, kita mo, sarap, steak, fried chicken. It will already stimulate your brain cells, and the brain cells will stimulate your stomach. So you feel hungry. Your mouth will what you salivate. Yummy. But if there is no cell-to-cell communication, then para kang zombie, no? Para kang robot. There is no interaction whatsoever. Okay? So this is the role, a very important role of the cells. cellular activation and communication through extracellular and intercellular signaling. Okay? So, try to please remember that there should be communication, activation through the signals received by the different cells in your body. Whether it is extracellular or intercellular signals. So, signals that most respond can be classified into several groups. Ayun. What are the signals? Damage, contact with the neighboring cells, contact with the extracellular matrix, and the presence of secreted molecules. For example, in this room, if I tell the President, Mr. or Miss President, please communicate to the class that we will have an exam in 20 seconds. So the class president, oh, one up sheet of paper. Okay? So that is the role of that cell. Siya ang parang DJ, no? Pumunta ka ng concert ng Journey na diba? Sa Coldplay. Very sweet. Taylor Swift, went to Taylor Swift concert. Eros, wala pa, Eros. Ayan, signal. Okay, signal. Oh. I can see now the expression in your face. Looks familiar. Meaning to say, you are actually now activating your cells in your brain. Okay? Now, what are the different types of signal pathways? You have the paracrine, autocrine, synaptic, endocrine. Okay, explain. Who wants to explain? Okay, right.
Ang autocrine po, Doc, is pag ma-release ng signal ang cell, the same cell ang mag-receive ng signal. Ang paracrine kasi, pag mag-release ng signal ng cell, nearby cell lang din ang mag-receive ng signal. Tapos endocrine kasi, once mag-release ng signal ng cell, malayo ang kanyang mapuntahan na target cell. Ang synaptic po, ang neurons, sorry. So, ang synaptic signaling po is between neurons. So, it's a neuromuscular junction.
Neuromuscular junction. Kinuro kita. Sabi mo, sige. Okay. So, those are self-explanatory. Now, we have the nature of the cellular stimulus paracrine. The cellular receptors and cellular surface receptors, you have the open ion, etc., etc. So, these are all involved in the signaling pathways. And again, they play a role in motion, communication, and even nutrition. Okay? Imagine if your brain cannot communicate with your mouth or your stomach. When you chew your food, naturally you have to swallow it. There's no communication to the what you call this, stomach. Then you will be chewing and chewing and chewing the food in your mouth and not swallowing it. Okay? You see now the connection? Muya ka lang ng muya. But without swallowing. Okay? So, what are the signal transduction pathways?
Signal transduction pathways would start with a receptor either on the cell membrane or inside the cell, and then these receptors would then continue to transfer the message through secondary messengers. So some example of signal transduction pathways would be the CAMP mechanism or the IP3 DAG mechanism, tyrosine kinase, things like those. So. What? I didn't say any cycles. Those are examples.
Okay, so here again would be the schematic diagram. Puro mga diagram, no? And then so you have the transcription. What is transcription? What do you mean by transcription?
For DNA transcription, usually i-transcribe na yung transcribe, i-interpret i-interpret na ng for example, DNA polymerase, so i-interpret na niya yung mRNA that will wait, no, the RNA?
mRNA.
During transcription, the DNA will be processed by RNA polymerase for it to produce RNA.
So, you mentioned messenger RNA and DNA. The messenger RNA plays a big role in the cell, okay? And the messenger RNA is usually located where?
Also the located
You need to find the RNA.
In the nucleus po.
cytosol.
Ay, cytoplasm po.
Cytosol man yata.
RNA po lang. In the cytoplasm po.
Cytoplasm, di ba? RNA.
In the nucleus po.
Malito, no? DNA is in the nucleus, RNA is
cytoplasm.
We have the messenger RNA. What's the usual location of this messenger RNA?
So what is the usual location?
Messenger siya eh. Same messenger. What are the places that you
Sa ribos.
mRNA po are located po siya sa ribosomes.
Information from the mRNA is being translated to protein flow.
Anyway, let's continue. You have again the schematic diagram, puro schematic diagram ito. Okay. For cell cycle progression. And we have the growth factors. Okay, so we have now the entry of these growth factor receptors and the active RAS. And then we have the cell cycle progression. And now we have the receptors with kinase activities, with the RTKS, and we have the receptor proteins of the Notch family and the Wnt protein ligands, the G protein coupled receptors, what are all of these things, no? And then you have the nodular signaling proteins, hubs, the enzymes, nuclear transcription, active protein. So we have activation and inhibition again coming into the picture. No? There is the process of sort of waking you up in the morning and letting you sleep in the evening. It's especially done when you were still a young child. Sabihin ng nanay, no, tatay, magtulog ka na at 6 o'clock in the evening. So that you will grow tall. Then in the morning, they wake you up. Bangon na. So you go to school. So they activate you. Okay? Now, transcription factors. Okay? This has already been discussed. And you have the DNA binding lobing permits, specific binding to short DNA sequences. What are short DNA sequences, by the way?
Short DNA sequences are
Short na DNA.
What's the importance of the short DNA sequences? Sure. Who is very sure? I'm very sure it's a long, short DNA sequences. Those short DNA sequences. You have the long DNA sequences. Ano yung ginagawa ng long?
The long DNA sequences basically regulates or suppresses gene expression po.
Short.
They hindi ako sure na.
Research on that. No? Short. You have the long, you have the short. Anyone who can answer?
Short DNA sequences can act as primers. For example, in transcription or in replication, DNA polymerases cannot know if saan sila magsisimula ng replicate. So, yung short DNA sequences can act as primers on the long DNA sequences that really carries the genetic codes.
What are primers again?
Primers, Doc, are needed in order for the DNA polymerase, which is primer dependent, to know what specific sequence is needed to be replicated or transcribed. Guide.
Stimulators and primers. Okay, very good. Okay, so you answered it. So you have the growth factors and receptors. Okay, what is the role of these growth factors and receptors? They promote entry cells into the cycle, relieve blocks on cell cycle progression, prevent apoptosis, and enhance biosynthesis. At this point, let's have a 2 minutes break.
Nahuli na ta?